Hello everyone. My name is Parnian Rafei. I'm a master's student of clinical psychology at the University of Tehran. Unfortunately, I was meant to be there with you today in the Human Brain Project student conference, but due to some visa problems, I sadly missed this great event. And so, Human Brain Project education team kindly allowed me to present my presentation virtually. So that's why I'm here with you today. I'm going to present a study which I have conducted at the University of Tehran, titled Episodic Future Thinking Deficit in Cannabis Users, an fMRI study of neurofunctional alterations. So first, you may ask me, what is the episodic future thinking, which I have studied? Episodic future thinking is a cognitive function that allows us to simulate the future events, novel future events, which haven't happened yet. So it is also called mental time travel. And if you close your eyes and think about that tour that you're going to have from the city of Pisa tomorrow, I guess, and you can just simulate and predict all the details of the event. It is your episodic future thinking that is serving you. And it has the distinct neural substrates, and the key neural substrate and brain region that serves episodic future thinking is, let me just is the medial temporal lobe. It is the key region, but also different parts of the brain, including medial prefrontal cortex, precuneus, lateral parietal cortex, or lateral temporal cortex, are also associated with the function of episodic future thinking in our brains. Episodic future thinking has different functions, but we predicted that episodic future thinking may have some different functions in the context of substance use disorder, because it is closely associated with different cognitive functions such as decision making, planning, anticipation, and delay discounting, which are all very important, crucial neurocognitive functions in the context of substance use disorder. And so, why we chose cannabis as a drug of interest in this study is mostly because it's the most widely used illicit drug worldwide, and different other reasons, like it's getting legalized in different parts of the world. It is accessible and affordable and available everywhere. And also, there's a growing body of literature suggesting medical benefits of cannabis use. So I think it's very important to go through different potential deficits that this drug may bring about. Going through the method, we had 40 participants overall, which were 20 right-handed current cannabis users, which were diagnosed with moderate to severe cannabis use disorder by DSM-5. And also we had 20 healthy controls, which were matched in age, gender, and educational level with the cannabis users group. And also the age was between 18 and 40. And this is like the gender distribution of the participants in our study. And we had some baseline assessments, which was Edinburgh handedness test, to be sure, in order to make sure that all of our participants are right-handed. Information related to their drug use. We checked their anxiety and depression level to make sure that they're not going through any pathological mood and anxiety disorders, and they are in a healthy range. And also, we use the Montreal Cognitive Assessment, which is also called MOCA, to make sure that all our participants are in a healthy range of cognitive performance. We use a saliva drug test in order to make sure that all our participants are just cannabis users and they're not multi polydrug users, I'm sorry. And also it was a brief medical examination to make sure that our participants are not going through any psychiatric or neurological or physical problems at the time. And the materials we use is a Siemens magneton prisma 3 Tesla MRI scanner, and the fMRI stimuli which we have used for the episodic future thinking task was 10 Q sentences related to past events and 10 Q sentences related to future events, which we have normalized before employing them in the study. And also all these sentences were recorded in a female voice and presented auditorily to the participants. And these are the examples of our Q sentences which we have used. Like one of them was, imagine that you're dining at a restaurant with your family in the future. Or for the control condition of the fMRI task, we had the episodic memory, the past episodic memory task, which was like recall a past memory of walking in a park. So after these cues were presented during the scanning session, the participants had 15 seconds to recall if it was the past episodic memory condition, or construct a novel memory if it's the episodic future thinking task for 15 seconds with as much details as possible. And then immediately after constructing or recalling each episode, they had 5 seconds for rating the vividness of the episode on an MRI compatible response box on a Likert scale. And this is the overview of the fMRI task which we have used as the rest and the Q exposure, and then the rating part. And we had 20 trials. Each trial lasted 45 seconds. So going to the results, because of the lack of the time, I cannot just go more in depth to the methodology, but going to results, the participants in the normal healthy group and the cannabis user group differed significantly in their depression scores and MoCA scores. Although we used these tests to just include our participants, include the participants which are in the normal range of depression and cognitive performance, and we excluded those which were not in the range, but still there were like significant differences. And also in the behavioral results, the vividness rating that the our participants went through in the scanning session, the episodic future thinking vividness rates was marginally lower in the cannabis user group with this p-value compared to the non-user group. And also going through the fMRI results, you can see here we had this future condition here, the past condition, and an F test, a future subtraction from past. And as you can see here, these are the contrasts that we have. The non-user group, the healthy control group, had higher brain activations in the left cerebellum, the medial superior temporal gyrus here, occipital fusiform gyrus, and the lateral occipital gyrus. It means that, in other words, that cannabis users has decreased activations in these parts of their brain in the episodic future thinking condition. The left cerebellum, medial and superior temporal gyrus, occipital fusiform gyrus, and lateral occipital gyrus. And in the episodic memory task, the past retrieval task, which is here, we can see that cannabis users had decreased brain activations in their right frontal pole here and the inferior frontal gyrus. These are the mean activations of each group in each condition, as you can see here. And here you can see that the cannabis users had no higher activations in their brain compared to non-users. As a conclusion, we can see here that the cerebellum, which had decreased activation in episodic future thinking task in cannabis users, might stem from the reach area, which is a reach area in cannabinoid receptors. So that might be one reason. Also, cerebellum has vast connections with limbic corticosteroidal pathway, which is called the reward pathway, and is very important in addictive disorders. And also, cerebellum is reported to have structural alterations in chronic cannabis users. So this might also be correlated with the results that we had in this study. Also, medial temporal lobe, as I told you, is a key region for episodic future thinking and prospection. So it's an amazing finding that we saw that cannabis users had decreased activation in their medial temporal lobe, and also delayed discounting and future decision making, which are very important functions related to episodic future thinking, are regions that are served by medial temporal lobe, and also all goal-directed behaviors and planning for future actions based on imagined outcomes. Also, lateral occipital cortex, which is named to be the mind's eye, which is quite crucial for imagination and mental imaginary, had decreased activation in cannabis users. And lastly, occipital fusiform gyrus, which we all know that region of the brain for vision-based cognition and face recognition, had decreased activation in cannabis users. And this part is also reaching cannabinoid receptors, and it is reported to have decreased thickness in cannabis users in other studies. So it is quite it's worth noting. And lastly, I know I didn't have enough time to go through all the details of our study, but you can reach me in these two emails that we have. Don't hesitate to ask me any questions. Don't hesitate to tell me your ideas. And unfortunately, again, I'm not there to discuss all my findings and hear about your findings of your studies. And but at least I was there to present a part of my research there with you today. This is the University of Tehran, where I study, and also, unfortunately, I cannot be with you today again, but science has no borders, so maybe next time. Have a very good day, a very good conference, and looking forward to hear your comments or thoughts on my presentation. Bye.